Welcome. This is Eduqas A-Level Biology, explained clearly and exam-focused. Let's make this make sense.
How can one tiny sugar molecule possibly provide enough energy for something like a marathon? The scale just seems impossible, right? Well, the answer isn't magic. It's just some really cool biochemistry. And that's really the best way to picture the Krebs cycle. It's a tiny but powerful engine. It takes the fuel that's been broken down from your food, and it systematically pulls out all the energy, getting it ready for the final big power-up. So, before we can see how this little engine actually runs, we first need to get our bearings. What exactly is it, and where in the world do you find it? Okay, here it is. This is the formal textbook definition, the one you'd want to have in your back pocket for an exam. And trust me, every single word here matters. From enzyme controlled, which tells us it's a super precise process, to oxidized, which is the chemical key to unlocking all that energy. And speaking of exams, these are the non-negotiable keywords. You have to know them. A really important one here is oxidation. In this context, it just means a molecule loses a hydrogen. And on the flip side, when we say something like NAD is reduced, it just means it gained that hydrogen. Simple as that. And this whole thing happens in a very specific spot. The location is absolutely key. The cycle runs in the mitochondrial matrix, the very center of your cell's powerhouse. And this is just brilliant biological design, you know? It keeps all the enzymes and products concentrated right next door to where they're needed for the next stage. It is all about efficiency. All right, so every engine needs fuel and a few key parts to get started. Let's take a look at the essential ingredients you need for just one single turn of this molecular engine. Here are our inputs. You've got acetyl-CoA, that's our main fuel. It's a little 2-carbon molecule that comes from glucose. Then you have NAD and FAD. Think of these as empty taxi cabs, just cruising around waiting to pick up some high-energy passengers. And then there's oxaloacetate. Well, that one's the real star of the show. And this, right here, is the absolute secret to the whole process. Oxaloacetate isn't used up. It's both the start line and the finish line. It's there at the beginning to grab the fuel, and then poof, it's right back at the end, totally regenerated and ready for the next molecule of fuel. That's what makes it a true cycle. Okay, we've got our fuel, we've got our parts. It's time to pop the hood and watch this engine run. We're going to follow the carbons as they go through what is basically a molecular roundabout. So first up, our 2-carbon fuel, acetyl-CoA, joins up with the 4-carbon oxaloacetate. 2 plus 4 gives us a 6-carbon molecule called citrate. Now the dismantling begins. In 2 quick steps, a carbon gets snapped off and released as carbon dioxide. Yep, that's the very same CO2 you're breathing out right now. And each time a carbon snaps off, one of our NAD taxis swoops in and picks up a high-energy hydrogen. And just like that, we've hit a huge milestone. The 2 carbons that came in as fuel, they're gone, completely dismantled and released as waste. But the process is not over, not even close. The rest of the cycle is all about grabbing the leftover energy and hitting the reset button. So what happens next? Another one of our energy taxis, FAD, comes in and grabs a hydrogen. Then something cool happens. A single molecule of ATP is made right there on the spot. It's like a little spark of power coming directly from the engine itself. And finally, after a bit of molecular shuffling, that original 4-carbon starter molecule, oxaloacetate, is perfectly reformed, ready to go again. Okay, so after all that complex chemistry, what did we actually make? What's the final output from just one spin of the cycle? Let's do the tally. 3. We get 3 molecules of reduced NAD, and this, my friends, is the biggest prize. Think of these as 3 fully charged power banks just waiting to be used. We also get 1 molecule of reduced FAD. It's another type of energy carrier, a different model of power bank, but it's also packed with potential. And we get just 1 single molecule of ATP. This is a tiny amount of directly usable energy. And you know what? That fact is probably one of the most important things to understand about this whole process. Now hold on, because we have to double everything in that first column. Why? Because a single molecule of glucose actually provides enough fuel for 2 turns of the cycle. So the real payoff from 1 glucose is 6 reduced NAD, 2 reduced FAD, and 2 ATP. So this brings us to the big picture question. If this cycle produces so little direct ATP, why is it this super important central engine of our metabolism? What is the real point of it all? Here's the most common misconception, all cleared up. The Krebs cycle's main job isn't to make ATP directly. Its job is to load up those coenzymes, the NAD and FAD, with high-energy electrons. The little bit of ATP it makes, that's just pocket change compared to the massive potential that's now stored in those energy carriers.
So yeah, the main job of reduced NAD and FAD is to be delivery trucks. They transport all that harvested energy to the final stage of respiration, a process called oxidative phosphorylation. And that's where the real ATP party happens.
Okay, here's a classic question that trips a lot of people up. We've walked through every single step, and we haven't mentioned oxygen even once. So why on earth do we call this an aerobic process, which means it requires oxygen? It's because all these processes are completely dependent on each other. The Krebs cycle can only keep running if it gets a steady supply of empty NAD and FAD taxis. The only way for those taxis to become empty is to drop off their energy at the next stage. And that next stage absolutely requires oxygen. So no oxygen means the taxis get clogged, a huge traffic jam forms, and the entire Krebs cycle engine seizes up. All right, let's nail this down. Just to recap, here are the big mistakes to avoid. 1. Saying the Krebs cycle produces lots of ATP. It doesn't. 2. Saying oxygen is used directly in the cycle. It isn't. And 3. Forgetting that oxaloacetate is regenerated. That's the whole magic trick that makes it a cycle in the first place. So our engine has dismantled the fuel. Our high-energy taxis are fully loaded and ready to go. The big question left is, where's the final destination? Where do they go to cash in all this carefully harvested energy and turn it into the power that runs literally everything in your body? Well, that's a story for next time.
If this explanation helped, subscribe for more clear Eduqas A-level biology videos.